This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, episode 1301, recorded on February 27th, 2026. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Montreal, Canada, Angela Mingarelli.
Hello, how's everyone today?
You get warmer? Is it warmed up a bit, or it's still freezing up there?
Well, so it's still freezing. It's still below zero, at least Celsius. But within the last 2 weeks, it's gotten a lot warmer. So it was like minus 15, minus 20 for over a month. Yesterday it was minus 14. What's that in Fahrenheit? Because if not, people are going to kill me.
No, I like Fahrenheit. It's
minus
good.
14. I don't know. That's like, what would that be? Around zero? Is that less than zero? I don't know.
Yeah, well, minus 0 Celsius is 32 Fahrenheit.
Exactly.
So it's not going to be
so
less
it's going to
than
be like
0 Fahrenheit.
5, I guess.
Maybe.
Something like that.
But
and yesterday
cold.
I didn't even wear a scarf. Minus 14 felt warm. I wasn't even wearing a scarf. I had my coat open and I was like, this is nice. But today it's only minus 3 right now. So that's like 27,
Spring.
28 something, which is warm. Warm.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 54 Fahrenheit, sunny, and that's 12 degrees Celsius. So
pretty
quite
mild
a bit
there.
warmer.
It's
Yes.
very nice. Is there any snow left on the ground?
Just little tiny patches in, north-facing buildings and stuff. Yeah.
We had a lot of snow. Let's see when. Sunday night, Monday, it snowed like 16 inches. And so everything was closed Monday. University was closed. Went back Tuesday. And then Wednesday morning, it snowed again, like 3 inches. And it was just such a mess. I drive like 4 miles to the train station. It takes me 5 minutes. And it took me over a half hour. I almost missed my train because people were going so slow, hadn't cleaned the roads. But now it's 4 degrees Celsius. It's sunny today. It's really nice. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. It's not freezing here. As a matter of fact, get this, it's 87 Fahrenheit.
87? I'm so
87.
jealous. Oh my God.
It's crazy. And so we're having a little warm spell here. This is, I think, unseasonably warm even for Austin. We're going to be in the 80s for another several days. Oh, geez, wow, for another week. Then we return to the 70s-ish. But sunny and stuff, I'm not complaining. It's good. Good.
So I wasn't in yet, but I wanted to comment on Angela's comment about how suddenly, a cold temperature feels warm. And we always say, and if it were November, we would just be crazy putting on mittens and hats and scarves and everything, or vice versa. In November, we're like, oh my gosh, it's so cold. And if it were this temperature in March, we'd be walking around with no mittens, gloves, or scarves. Yeah.
I can tell sort of my weather person is the cat. Because below a certain temperature outside, the cat sleeps on the bed. And I don't know what the cutoff is. I have to do the measurements. Okay, but other times he's on the floor by the bed.
For a couple of weeks, we were at like minus 10-ish all the time. And I know, and then when it finally got to 0, it's a big difference in how you feel, right?
Oh, yeah.
Minus 10 really hurts, and you have to bundle up. But at 0, you almost don't need a scarf or a hat or something,
I almost thought about not wearing a coat today because it was minus 5. I saw people though wearing just sweaters. Like in Montreal, from what Kathy was saying, it was so cold for so long, minus 20, minus 25, that minus 5 feels like spring weather.
If you enjoy these programs, we'd love to have your support to support them. I should find a different word, right? We'd love your support to produce them because they do cost money to produce. MicrobeTV is a 501c3 nonprofit. So if you donate money, you can deduct it from your federal, or you could try to deduct it from your federal tax returns. And it helps us to produce these programs. MicrobeTV/contribute. I want to remind you that Rosenfeld Lab is in Philadelphia now. They're looking for a postdoc and a tech to work on enterovirus pathogenesis and immunobiology, things like the function of the cross-reactive or the implications of the cross-reactive antibody response, making mouse models for these viruses, and developing new vaccine platforms. Amy's email, it's Amy Rosenfeld, of course, at rosenfelda@mlhs.org. Okay. We have a couple of interesting papers for you today. The first snippet is it's a early release. It's been reviewed and so forth. And it's in Emerging Infectious Diseases, which is a journal of the CDC. And for a while, we wondered if it was going to continue, but it's continuing, which is
thank
good.
goodness. And this, and it seems to be intact. This is one of my favorite journals because they're a quick read and always of, current interest.
Yeah, these are good for snippets.
Yeah.
But they're very interesting.
Yeah.
So this one really caught my attention. This is lymphocytic choriomeningitis virus seroprevalence among urban pregnant women and newborns, Philadelphia, Pennsylvania, USA, 2021. It was just published in March. Well, it's coming out in March, right? It's ahead of publication, but the study was done back in samples from 2021, as you will see. So lymphocytic choriomeningitis virus is a rodent-borne pathogen, and it is well known among virologists because it is a workhorse for immunologists who use it all the time in their studies. And I remember many, many years ago at a meeting, there are always talks on LCMV or the use of LCMV in immunology. And after one of them, this herpes virus guy, Kathy, what's his name? Barry something? Do you remember his name? Barry.
Virus. Where is he from?
Tennessee, something like that.
Sorry, not coming up with it.
Anyway, he was always sarcastic. And then he got up and he said, all right, now I'm going to talk about a virus that actually causes human disease. But he was wrong because this one causes human disease. But that's the kind of snark that he did. I don't remember his name.
So, Vincent, your lead-off sentence, it's like you're channeling exactly what I wrote at the beginning of my summary.
Okay, I'm sorry.
Go
ahead
right.
with your summary.
It's a virus that we know because of its utility in studying viral immunology. However, it causes severe congenital infections. In adults, it's mostly associated with asymptomatic or mild illness, except in immunocompromised patients. In fetuses and newborns, it leads to a number of things. One is choriomeningitis, which is a pretty bad eye complication, neurological malformations, intellectual disability, neurodevelopmental problems, and fetal death. So, as Vincent said, it's rodent-borne. It's carried by the house mouse. It's found in urban and rural settings throughout the world, although they don't report anything for Africa or Antarctica, but all the other continents are mentioned. However, there's not much epidemiological data, and so they wanted to test how prevalent it is. They tested 3 populations. One was just a random sample of women who gave birth in Philadelphia. Another was a population that they called high risk because it was from women who gave birth also in Philadelphia, but who came from places with a high, quote, neighborhood deprivation index score. I didn't go through and look at what that is, but I think the idea is that with higher neighborhood deprivation, there would be more rodents and more possibility for exposure to them. And the third group that they looked at was a group with prenatally diagnosed neurological malformations. And they tested serum samples from the mother and in some of the cases from paired cord blood collected at birth. And so they were testing several hypotheses. Did seropositive people have evidence of acute infection at the time of collection? And the answer to that is no. Was there transfer of antibodies from the pregnant mother to the newborn? The answer is yes. How common is LCMV? They call it common. somewhere between 2.4 to 2.7%, which matches some other previous sero surveys. Would people in census tracts with higher neighborhood deprivation scores have higher seroprevalence of LCMV? The answer to that was no. And then, were congenital malformations related to congenital LCMV? And the answer to that is no. And the caveats are discussed, and we'll talk about more of those too. But although they start with a large population, when they get down to some of these subgroups, their numbers are not quite as big.
Can I also add that a risk factor? So as you said, it's in rodents. And we know now from immunology, obviously, because I'm in an immunology lab and we use a ton of LCMV, that some of the studies that have been done on my floor in other labs, people are using pet shop mice. And all of the pet shop mice that they got are infected with LCMV. So be aware that pet shop mice are very dirty. We use them as like a dirty mouse model, and they have everything. Like, they won't even let us bring them into our animal facility. So just be aware that if your kids have rodents, if you're buying them like little mice as pets and you're pregnant, that they could have LCMV.
Also, hamsters too have
Exactly.
LCMV.
Yeah. Also like a cute home pet that could potentially be dangerous.
Or
So,
if
Angela,
you have snakes and you buy mice for your snakes.
Also.
Right.
Exactly. Yeah.
Angela, why is it such a good immunology model?
Well, so Rafi Ahmed would say many things, T-cell immunologist, but so it's good because in LCMV, there's two different viruses. So there's LCMV Armstrong, which is an acute infection. And then the same virus with one amino acid change is LCMV clone 13, which is a chronic infection. So you can have a chronic model and an acute model, and it infects all tissues. So it infects, I'm not sure in humans though, this is in mice. I think, well, at least in humans, we know that it infects the eyes and the brain from what Kathy's summary said. But in mice, it infects spleen, liver, lung, gut, brain, salivary glands. Like, it infects everything. So if you're studying, for example, T cells, or you want to study memory T cells and things like that, exhaustion, especially in chronic antigen exposure, like in clone 13, there's just a lot of things that you can do with LCMV, especially with T cells. And it's easy to inject, like clone 13 is IV, Armstrong is IP. And they're BSL-2, which is also nice that you don't have to work in like a BSL-3. Yeah.
So
I don't know if you said this, but a big thing is that the virus is non-cytolytic, so all the pathology in an animal is immune mediated.
Exactly. Okay. Also, and now there's even a lot of models, if you've ever heard of like P14 mice or SMARTA mice. So P14 is a TCR transgenic mouse that's specific for GP33 to 41 peptide. So it's a specific epitope in LCMV. And then, so all of the T cells are specific to that part of to GP33. And then SMARTA is for CD4 T cells. So then you can track T cells. People do adoptive transfers and things with like antigen specific T cells. We do that all the time. With either P14, so CD8s, or SMARTA, CD4 T cells. So yes, there's lots of tools for LCMV and immunology.
Are we concerned as laboratory workers about is there a hazard working on this in the lab, and what's the story?
Yes, my PI actually told me that she knows of a man that was hospitalized with an LCMV infection. It was unclear if he had stabbed himself or if he had acquired it via a mouse bite. I forget how it went. I don't remember exactly how, but I know that he was hospitalized with LCMV. He was also slightly immunocompromised, and which also question as to why was he working with LCMV. But anyway.
So
yes.
You mean needle stick?
Yeah, because you do IP normally, intraperitoneal, so you're using needles when injecting. So maybe it's like it is possible to stab yourself accidentally.
Yeah, of course. It's just funny that you use stab.
Oh, sorry. Yeah, I should say inject.
It sounds like he was trying to hurt himself.
I should say inject. We use these words in the lab like colloquially. But no, there's of course a risk. And if, yes, a woman is pregnant, then obviously she shouldn't be working in the lab. Apart from the isoflurane and things like that, the actual viruses are dangerous. So yes, there is a risk, which is why we do everything in BSL-2. And the mice are also housed in BSL-2+. So, yeah.
So the guy's name is Barry Rouse. Does that ring a bell?
Okay, yeah, sure.
Yes.
Yes.
And you mentioned Rafi Ahmed. also it goes way back to Mike Goldstone at
That's right.
Scripps. So I'm trying to figure out.
That's probably
How
where
far?
Lindsay Witten got it from.
Yeah.
Goldstone.
Yeah.
Right.
I'm pretty sure, I forget the paper, but they defined T cell exhaustion using LCMV, that model specifically in T cell. So like it's helped the T cell field a bunch.
And so just to clarify, the congenital infections have to do with a mother being infected and that infection crossing the placenta into the fetus, correct? And those are the really nasty ones in terms of neurological defects.
Exactly, because the developing fetus already has the virus in it as it develops. And
Right.
it was interesting though, like, well, I guess we have to get into the paper and into the snippet, but I think they measured in 37 women that had either some sort of brain malformation or I forget how else they defined it, but fetuses that had some sort of neurodevelopmental issue, and all of them were LCMV negative.
Yep.
So it's not necessarily like Kathy said that if you have LCMV that you're going to have some sort of malformation in neurodevelopment.
But you can.
You can, yes, you can. You can, of course. Avoid rats and mice.
Some early exhaustion papers come from also Zinkernagel, Ralph Zinkernagel. So, yeah.
Okay, so house mouse and hamsters, pet mice.
Also pet rats. People do rats now
as
too
well.
as pets. So,
all right. So as you heard, it causes severe disease in humans, immunocompromised patients, but has also effects on fetuses and newborns when you get infected during pregnancy. And you can get all these diseases that Kathy enumerated: chorioretinitis, intracranial calcifications, a variety of neurologic malformations, intellectual disability, neurodevelopmental problems, and fetal death. So they actually say LCMV is a devastating congenital human pathogen, but there's not a lot of epidemiological data describing domestic seroprevalence in the U.S. So there have been some previous serosurveys in the 80s and 90s that found this LCMV seroprevalence of 2 to 5%, which means 2 to 5% of the people they looked at had antibodies to the virus in their serum, right? 2 to 5%. That's not insignificant because we have many millions of people here in the U.S., right? They say it's suggesting exposure might be common. In 1984 to 89, in Baltimore, Maryland, 9% of house mice had detectable antibodies, and infected mice were identified at 6 of 8 sites, and the seroprevalence of mice varied from 4 to 13%. The site that had the highest seroprevalence was an inner city residential area, where the positive mice are clustered within city blocks and households, and clustering correlated with estimates of mouse density. So, all right. So these infants with neurological malformations, many infants are born with them, and but most, they say 50 to 60%, are idiopathic, which means we don't know what's causing the disease. And one previous survey in Chicago showed that children with congenital chorioretinitis had the most common factor was LCMV exposure. So it's a possibility that LCMV infection is underdiagnosed. So that's why this study came up. They wanted to look at LCMV seroprevalence among a high-risk sample of pregnant women in Philadelphia, Pennsylvania. So they are using available, what they call remnant serum samples. It's like when you go to a store and you buy remnant cloth, right? They're going to make some cloth remnant serum samples from pregnant women and newborns at 2 birth hospitals, Pennsylvania Hospital and Hospital of the University of Pennsylvania, and a freestanding Children's Hospital special delivery unit. That's all in Pennsylvania. So as part of what happens when you're going to have a baby, they take blood they test for syphilis, actually, and then they throw it away. And so these people took the blood that was going to be disposed, and it's all the identifying factors are taken away.
Shouldn't they also be testing for toxoplasma? Or is that not regular now? Do we know?
Well, here they only said syphilis, so I don't know.
I think they do all that torch testing way earlier in the pregnancy.
Okay, true. Yeah.
Yeah, this is at admission for delivery.
Okay,
right?
True.
Yeah. So they got they also got, in addition to these remnants, they got samples from pregnant women who have referred to CHOP, Children's Hospital of Pennsylvania, a special delivery unit for childbirth because of known fetal neurologic malformations. Okay, so they have a repository for that. So the study is done in 3 phases. Phases 1 and 2, they have serum samples from pregnant women who were admitted for childbirth from January 1 to December 31st, 2021, so the whole year, and for which they had an available remnant sample. And they also got remnant cord blood samples, right, to see if the fetus had antibodies as well. So, and they wanted to see if the seropositive women transferred antibodies to the fetus. So for phase 1, they defined what they call a high-risk group, where they assume there's an association between living in a neighborhood of low socioeconomic status and higher mouse exposure. They assumed that. I don't know if it's actually true. I mean, nobody wants mice in their house, no matter how poor you are, right? So people try and keep them out. I mean, mouse traps are not very expensive, I guess. Anyway, so they have what they call, there's a neighborhood or a community deprivation index, as Kathy mentioned. And so they selected samples from pregnant women who lived in these census tracts with the highest quartile of neighborhood deprivation. Their incomes are below poverty level. They have at least a high school education. They lack health insurance. They get public assistance for income or food. The income is low, and also the neighborhood. So this is how they characterize the neighborhoods, by these criteria. And also there are a fraction of vacant homes in these neighborhoods, right?
So there could be more rodents in the vacant homes.
Yeah, that's right. For phase 2, they wanted to get a seroprevalence estimate for the study population at large. They selected serum samples from the women who gave birth during the study period. And then phase 3, they wanted to look at the LCMV seroprevalence in this cohort of women with known fetal neurologic and ocular malformations. All right, so they collected all these samples and they sent them to the CDC. Boy, I guess collecting is the hardest part. And the CDC did an ELISA for LCMV IgG antibodies. And then the ones that were positive for IgG then looked for IgM, which would mean closer to the infection time, right? Because the IgM antibodies come up first. And then they extract RNA from these IgM positive samples, because those are the ones most likely to have virus in it. And then they do PCR, RT-PCR, to look for LCMV. Okay, so here are the numbers. In 2021, the two hospitals reported that 9,286 women gave birth to 9,502 newborns. So a couple of moms had more than one baby, which happens. And of the 700 selected maternal samples from this phase 1, 17 were positive for IgG. That's 2.4%. And none of those were IgM positive, so not really recent infection. And they also had 11 paired cord blood samples from these 17 IgG positive maternal samples. They were all positive for IgG to LCMV, but IgM negative. So they matched the moms.
Yeah, so it's a thing that even a virologist who doesn't know very much immunology like me knows that IgM is characteristic of pretty much an active or just very recent infection.
Yeah,
and they were negative for that.
Mm-hmm.
Phase 2, 8 of the 300 maternal samples were IgG positive for LCMV. That's 2.7%, so very similar. None of them were IgM positive. 6 paired blood samples, all were IgG positive and IgM negative, so similar to the first. They've compared then the serostatus with clinical and demographic characteristics for phase 1s and 2 combined, and the statistically significant factors were the birth hospital, so one hospital ranked higher than the other, the maternal race. All races except white were higher. Well, they're not really races, right? Because there's no such thing as race.
Right.
It's ethnicity. Why does everyone still use race? I don't understand. I remember it's historical.
Mm-hmm.
Yeah.
Okay. And maternal ethnicity, Hispanic ranked higher than non-Hispanic. Right? So that's phase 1 and 2. And then phase 3, they have this cohort of women carrying fetuses with prenatally diagnosed congenital malformations. And so they tested for LCMV IgG in the plasma from 37 pregnant women and cord blood samples from their 39 affected children. And some of these malformations, microcephaly, 17, ventriculomegaly, corpus callosum abnormalities, encephalocele, retinal hemorrhage, Dandy-Walker malformation, absent septum, pellucidum, and posterior fossa cyst. None of them were seropositive for LCMV.
Not big numbers. There were
No,
only
they're
37, only 37 women.
Yeah, I mean, if you got 2.5%, you're not going to really pick it up there, right?
But my question is, they talk about ethnicity, but they don't talk at all about comorbidities, and if the people that had LCMV infection were possibly, I don't know, diabetic or had a chronic condition or something else that could have been a factor, which I think could be interesting. I just don't know how available that data would be to them, or those data, sorry.
Yeah.
No, it's true.
They did say that, they did get some of these data, like where they lived and that kind of thing, and then they de-identified it, and that de-identification happened before they even tested the serum to make it even more, distinct or distant from. But so I think they just collected certain bits of information.
So that's the data, and what do we learn? We learned that 2 to 3% of a cohort of pregnant women in Philadelphia had past exposure to LCMV. In fact, did I say pregnant women? 2 to 3 per 100 pregnant women, and they also passed the antibody on to their fetuses.
Maybe their comorbidity or risk factor was that they worked in labs. Imagine they're like in immunology labs. Well, I mean, the thing is that I think for us, we must all have titers of, like, we must have been infected at some point, even if it's just, yes, we work in BSL-2s, but I wonder if you did this study in people, in immunologists, if it would be 100% or not.
That'd be fun.
Maybe.
Yeah, it's an interesting point.
Fun.
I don't know if they asked them that.
Yeah.
But, all of us who worked on viruses have high titers of antibodies to our virus. I used to have high titers to polio. I probably still do.
Yeah.
I'm sure Rich Vaccinia, Kathy Adno, and Angela LCMV.
Yeah.
So this aligns with previous sero studies in urban Baltimore, right? Similar percentages. So that means in Philadelphia, between 30 to 45,000 people have been exposed, including 400 to 600 pregnant women. So exposing this, supports the idea that exposure to rodents shedding LCMV is common. So we would like to develop guidance for pregnant women to avoid rodent excreta to the extent possible. So if you're in your house and you find rodent excreta, which I do all the time, right, in the basement, and I always tell the virology students this: don't just sweep it up, because that's going to aerosolize it. Just put bleach on it, right? A dilute solution of bleach, and let it inactivate for a while, and then you can sweep it up.
Maybe wear a mask if you've got one.
Yeah, a mask would be great. It doesn't even have to be a fancy mask. Just a mask would be good.
I had a friend who found dead mice in his outdoor grill, and
I find that
before you clean it up, put a mask on.
Imagine you made a steak.
I find that every spring before I turn it on the first time, I have to make sure there are no mice because I don't want to fry mice or grill mice.
Yeah. Is this a cold weather climate thing? Because actually, I guess we used to get mice in California. Well, I don't remember.
Well, that was somebody in Santa Fe, but it does get colder in the winter. But this was in the summertime that they were alive. And yeah.
Because in Florida and Texas, I have not, it's not entirely true. There have been.
Well, I guess they're not trying to, especially in the winter, the mice in Florida and Texas, they could just be outside and they're fine. I think here they have to find shelter. So they're
probably happy it's winter.
Yeah.
I don't know. Do mice get happy?
I mean,
okay, so this is a very interesting question. So years ago, we did a paper, and you, Rich, you should remember this, where and the picture for the image was with mice. Mice all look the same as happy, sad, angry, frustrated, this, that, and the other. And they were all looking the same because in this study. But then someone last week, I don't even know where it was saying about how mice have different facial reactions. But I never saw a mouse face change.
It's true. So in like the veterinary aspect of working with mice, there is something called the grimace scale. So
that's right, the grimace scale. Yes,
exactly.
Yes.
So but it is true. So if you see a mouse with like its eyes kind of shut, it can mean pain, or if their lips are like more pursed, that their hair is a certain type of way.
Okay,
it's mostly pain though. Yeah. Pain. And
so this was
like
we did a paper on TWiV where they took fecal bacteria, feces from people with fibromyalgia, right, chronic pain, and they fed them to mice, and they measured their pain indices, and one of them was facial grimace. Yeah, but it induced pain in the mice.
That's crazy.
Isn't that crazy?
Yeah.
Feces from humans induced pain in mice?
Yes.
So like it's a microbiome effect?
It's a microbiome effect.
That's crazy.
We take it with a grain of salt because I don't know.
Has anyone ever tried to like deplete the microbiome of people with MS to see if you reconstitute it with other probiotics and other things, it could improve outcome? Is that a thing?
I'm sure it's been done. I don't know.
Okay. I have to look
into that. That's where I heard about the facial grimace, and
interesting.
I was surprised at that. Anyway, so as they say, there's already guidance for pregnant women for other pathogens: listeria, cytomegalovirus, toxoplasma, syphilis, rubella, varicella, Zika, and others. There's a lot. Basically, stay home.
Yeah. Don't do anything.
A lot of danger out there.
Whatever you do.
And the other thing that they thought was that the people residing in these census tracts with higher neighborhood deprivation would have higher rates, but they didn't. There's no difference between people from the higher places.
That's one of the most interesting things about the whole study, I think.
Well, and also the congenital malformations didn't correlate with higher titers.
Yeah, that's right. They say because it's a small sample, which could very well be. Anyway, so there you go. Now, I hope you've learned a lot about LCMV, folks, because we don't really talk about it that much.
I'm not sure we mentioned the authors. And so
we didn't. Go ahead.
Okay. Dustin Flannery is the first author, and he's at Children's Hospital. And Caitlin Kosoboom is a co-first author, and she's at the CDC. And then there's a bunch of middle authors from some of those places, many from CDC. And Scott Gordon is from University of Pennsylvania Perelman School of Medicine. So that's summary of the authors. So Philadelphia and Atlanta.
Now we're going to switch to amoeba. You should stay with us. Okay, this is pretty interesting, very interesting.
And don't get lost because at least one of the picks could almost be a snippet, one of the picks of the week. So
yeah, I heard. Okay. So before I do the paper, so the paper is about a giant virus infecting amoeba, and every couple of years I go to the giant virus meeting in Germany, and Rich came one year actually, and it's run by Matthias Fischer, and so this year he sent me an email after he listened to that episode. I just finished listening to the whole episode. Hope your audience will appreciate the more exotic viruses and hosts discussed. It's great that you keep highlighting viruses outside the clinical spectrum, as it really shows how ubiquitous and diverse viruses are and how they affect life on this planet in so many ways. On the other hand, I hope that it also becomes clear to the listeners that we cannot study every single group of these viruses in as much detail as has been done over decades for many human-animal pathogens. Mostly this is due to cost-benefit calculations and the overall research goals, which differ quite a bit between the clinical and environmental flavors of virology. On the former point, developing molecular tools for these exotic eukaryotic hosts is far from easy. So at the meeting, they talk about giant viruses and all kinds of mostly hosts in the ocean, unicellular hosts besides amoeba and plankton of various sorts, algae. It does not make sense to spend years of labor and funding to achieve genetic manipulation for a virus host system that is used just by a handful of labs worldwide. On the latter point, such detailed techniques are often not necessary to address questions of virus host coevolution or how a particular virus impacts its host populations in the wild. While some virology questions are clearly universally important, for example, characterization of viral genomes, particles, and replication cycles, others that are central for clinical models may not be so relevant in an eco-evolutionary context, such as identifying a particular receptor or developing drugs against the virus. Yet other aspects simply don't exist when working with unicellular hosts or are so poorly understood that there are no data, e.g. immunology. There will always be people who argue that we should disregard any viruses that don't make us sick, but this would restrict virology to a minute fraction of a % of all viruses out there. If we want to understand the various ways in which viruses have evolved and are shaping life on Earth, we have to keep looking in dark and unexplored areas. After all, science is about understanding the world we live in, and we may even learn some fundamentally new biology along the way. See Delbruck's phage group.
That's a very nice introduction. That's great.
That is.
So, LCMV is like a model. I mean, it's a mouse virus. Basically, it's a model system because it was established so long ago at the very beginnings, right? And so people started working on it, and it may not have been established today. Who knows, right? But we have model systems that are studied a lot, and then often very interesting viruses are discovered, but you can't study them because you can't grow them, as Matthias says. So this paper is really, it's about a giant virus, which is in the end a model system, but it addresses a lot of questions that people have had. And do you have a summary and stuff, Kathy? Or,
I do have a summary.
All right, let me do the authors first.
Yep.
So it's a Cell paper. Giant DNA viruses encode a hallmark translation initiation complex of eukaryotic life. And the authors are Maximilian Fels. Gosh, you got to love Maximilian.
It's a great
Isn't
name.
that great?
It's a great name.
Aidan Hill, Richard Hahn, Jasmine Garcia, Hugo Bicio, Chantal Aborgel, Phil Kranzuch, and Amy Lee from Harvard Medical School, Dana-Farber Cancer Institute, and the Aix-Marseille University in Marseille, France. And so Chantal usually goes to the giant virus meetings, but she wasn't there. And her husband there, who is, what's his name? Bergel husband. Let's see if that gets it.
So I was asking myself if giant viruses could infect humans. And I found on the internet that the first that Mimi virus was actually discovered. It was in an amoeba, as Kathy's going to say, but it was actually in a pneumonia outbreak. They identified this Mimi virus in England in 1992. That's when they discovered this Mimi virus, which was actually in an amoeba inside someone's lung. Interesting.
Yeah,
they thought it was a bacteria.
They thought it was a bacteria, so they put it away for 10 years.
Exactly.
And then Mimi comes from microbe mimic. So her husband is Jean-Michel Claverie, right? Who they both been on TWiV. And when I saw him at the giant virus, he says, "Oh, Chantal, she has a great paper. You're going to see it. You have to do it on your program."
Here we go.
So here we are. Jean-Michel, okay?
And this is a great paper. I really enjoyed this.
And it comes out March 2026, although technically it's February, although it says March, so we're ahead of the date.
Right.
Okay, Kathy, go ahead.
So, and it's an open access paper. Mm-hmm. So, virus infections impact a lot of cellular processes, And many of the things that viruses do are lumped into a term called host cell shutoff. So the viruses can affect host DNA synthesis, a lot of things regarding messenger RNA synthesis, like processing. They can result in degradation or decapping. And the host cell shutoff can also affect host translation. And all of these things provide an advantage to the virus to shut down the host and shift the bias of macromolecular synthesis toward itself. There can also be direct interference with translation. For example, near and dear to Vincent is the poliovirus infection, in which a translation initiation factor gets cleaved, and that's bad for the host messenger RNAs, but poliovirus has another way to get translated, so the virus has evolved this mechanism. Other viruses block entry of the messenger RNA into the ribosome, like SARS-CoV-2 and I think SARS-1 also. They can inhibit polyadenylation if their own messages don't need to be polyadenylated, and so forth. And some viruses encode molecules involved directly in translation. And so some viruses encode tRNAs. Since 1968, some phages have been known to do this, phage T4. Some proteins of the ribosomes themselves are encoded by viruses. So ribosomes, reminder, are made of ribosomal RNAs and ribosomal proteins. And so some viruses make cognate proteins for the ribosomes. And giant viruses, as you might expect, encode a lot of genes. And a lot of those genes are similar to cellular genes, and they're cellular genes that are not found in the rest of the virome. So we really wonder what a lot of those genes are doing. And some giant viruses are known to encode tRNAs, some aminoacyl tRNA synthetases, which are the enzymes that put the amino acid onto the tRNA, and some putative eukaryotic translation factors. And so in this paper, they're going to show us that giant DNA viruses encode a eukaryotic translation factor. And now we're going to get into a little bit of alphabet soup. So EIF4F is the factor. So you can remember F is for factor, and E is for eukaryotic, IF is initiation factor, and it's number 4. And the other factors that are going to play a role in this paper are the individual proteins that make up this F complex, and they are EIF4A, EIF4G, and EIF4E. So you just want to remember A, G, and E, they make up 4F. So they show that this giant virus encodes this complex that replaces the host EIF4F with its own, and so they're going to call it V viral EIF4F. And this then regulates viral translation, virion formation, and interestingly, they even show that it increases some fitness to environmental stress, such as starvation. And so I just have a couple of comments here. Because of all this past knowledge about translation, I kind of was a little bit put off by the title and also stuff that I heard in the popular press. So giant DNA viruses encode a hallmark translation initiation complex. I think they could have done away with the word hallmark. And then they add encode a hallmark translation initiation complex of eukaryotic life. And so I thought, okay, they're trying to come down on the our virus is alive or our virus is not alive. And so I thought, they were kind of pushing that a little bit. And then there are a couple things throughout the paper, but one of them is they talk about this being a fundamentally distinct form of translation. And I would take issue with that because I think we have other examples of things affecting initiation complex formation and capping and so forth. And so the basic mechanism of ribosomes transiting along a messenger RNA and producing proteins, I would call that the fundamental form of translation.
Yeah.
But,
Yes.
so, okay.
Agreed.
Anyway,
Agreed.
that's not to take away anything from, I think it's a really exciting, remarkable paper with really lovely experiments, logical, well thought. So, yeah.
So I didn't know this, but genes related to protein synthesis are the majority of genes in eukaryotes. In life, it's, they say, both prokaryotes and eukaryotes. So protein synthesis is really important. Well, duh, you have to make proteins, right?
Yeah.
Oh my gosh.
To me, protein synthesis is, of course, I cut my teeth on protein synthesis as a graduate student. I mean, an undergraduate. That's what got me into this. But to me, that's really the missing link in evolution. It's really hard to understand how we got from, even self-replicating nucleic acids to this extraordinarily complex machine that winds up translating the code. It's unbelievable.
What's interesting is that, as you know, the catalytic part of ribosomes is RNA.
Yeah.
And so that has suggested to many people that we had some kind of ribosome in the RNA world.
That's
right?
probably true.
Which we'll never know, I guess, but it's really interesting. So protein synthesis has evolved, right? So prokaryotes were first, and they have a protein synthesis machinery. And there are, in eukaryotes, which came later, there are defining adaptations. And one of them is this cap on the mRNA, which binds EIF4F. So there's a fundamental problem with protein synthesis, and that is you have to get the ribosome to the mRNA, right? And so how do you do that? Well, there's prokaryotes do it in one way. They actually have a sequence in the mRNA that's complementary to one of the ribosomal RNAs. This is called the Shine-Delgarno sequence. And eukaryotes do it by this EIF4G, which is a protein complex of 3 proteins, more actually, that binds the cap. And then the ribosome is recruited to that, and then it moves down merrily. So that's the background here. And now this paper is all about understanding why a virus genome would encode, or what's the function of a virus genome encoding a part of the translation system, right? Which they actually stumbled upon. I don't think they originally were looking at that, because other people do. For example, some giant viruses encode ribosomal RNAs, and people have shown that in a virus-infected cell, they get incorporated into ribosomes. But what it does, we don't know. It's there, but what is it doing? And this paper, I think, is the first to show what's happening. So it's very cool in that sense. Okay.
To your point, Vincent, I love thinking about when I see papers like this, I love thinking about the actual first experiment that led them down this train of thought. Because obviously it wasn't, like you said, they probably didn't go in with that question. But I always like to think about, out of all of these experiments, where did they start? And then how did they then bring it back home? Like bring it
it's always because they accidentally stumbled into this viral
probably
4G, as you'll see. But anyway, so they're working with giant DNA virus, virus, whose name is Acanthamoeba polyphaga, which is the amoeba virus. And by the way, they should not italicize it, which they do. If you use a virus which is named after the host, you do not italicize the name. Okay? Come on, folks. I don't even work on these viruses, and I know that. But I've talked to Jens Kuhn, and he says, Vincent, you don't italicize them. Anyway, so this virus infects the amoeba, and so they said, we hypothesized that a translation factor encoded by the virus would interact with ribosomes. Okay, so they purify what's called polysomes from virus-infected cells. So, an mRNA is associated with many ribosomes, not just one, and so it's called a polysome. And it can have 1, 2, 3, 4, 5, 6, whatever, right? And when you separate them on a sucrose gradient, you see all the peaks depending on how many ribosomes are associated with the mRNA. And so they use mass spectrometry. They do a polysome, which is ancient technology, and they use mass spectrometry to identify host proteins enriched, and they find some viral proteins there too, which have homology to EIF4A and 4E, which are associated with EIF4G complex, right? And they're sedimenting with these ribosomes. So they call them viral VIF4A and VIF4E. They put a V instead of an E, which stands for eukaryotic, right?
Even I'm gonna get picky here. They actually isolate from the polysome gradient the 40S fraction. Right. That is is not as big as the others. It's not even a monosome that has the whole ribosome on it. It has just the 40S subunit and the messenger RNA, because that's the initiation complex. Yeah. And after that advances, the initiation factor in question falls off. So if you want to look at that initiation factor in particular, you focus your attention on the 40S fraction.
Yeah, they have a nice picture of it here.
Yeah,
polysome. That's the fraction they have there. That's right.
This is one of these papers. This was easy for me to read. Of course, it's interesting because it's the kind of stuff that I like, that I'm used to, but it's got the current day technology associated with it. It's got old and new, right? We got co-IPs and polysome gradients, but we got mass spec and sequencing and all that stuff too.
Okay, so when they're doing the mass spec on the 40S fraction, they also find 54 associated viral proteins with no homology to any known protein. Okay, so what they do, they call this de-orphaning these proteins, which I thought was funny.
Yeah.
They do a structural homology search. So they use AlphaFold and another program called FoldSeq, where you can fold the proteins and say, does this folded protein now look, the predicted folded protein, of course, does it now look like some known folded protein?
Once again, there's the modern technology for you.
Yes.
AlphaFold is good enough so that you can take a sequence that you don't know anything about and fold it and say, oh, that looks familiar.
That I thought was so cool. Just being able to do that. Because if not, they would have had no idea what the 3D structure looked like. It didn't.
No, that's just the key here, yeah. Because the protein sequence is so diverged, but the structure is maintained, right? Anyway, they find a protein called, a viral protein, R255, which is a structural homologue of the middle domain of EIF4G. So EIF4G in eukaryotes is a long protein that binds a number of other proteins, including EIF4E, the cap binding protein, EIF4A, and EIF3, among others. So they say, so this is a structural homologue, and they say to validate this finding, we determine the crystal structure.
Yeah, I have that highlighted in mine.
Oh my gosh.
That's, for folks out there, that's funny because that used to take years, okay? And even then it was hit or miss. Oh yeah, sure, we'll just make crystals and get the structure.
So anyway,
I do appreciate, though, this paper, even though they do a ton of different things, their figures are very clear. For someone that, Rich, it might be easy for you to read. For me, I don't do experiments like this at all, so it's a little bit more difficult. But I find that the figures, though, are quite straightforward, and it wasn't too complex or deconvoluted. I feel like they did a good job of making it clear.
Yeah, absolutely. I was able to just kind of look through the figures and understand what was going on.
Mm-hmm.
Anyway, the structure analysis confirms that this is a structural homologue of the middle domain of EIF4G. So they call it VIF4G, viral IF4G. And they find these homologues in other giant viruses, but they're different. They're all different. So that's interesting in itself. So as I had said already, so EIF4G is a scaffolding protein basically for proteins that associate with it and make EIF4F. And then that is bound to the mRNA 5'N, and then the ribosome is recruited to that. So they asked whether VIF4G, it's very short, right? It's just the middle domain, could still form the 4F complex with 4A and 4E. And here they ran into some problems. They couldn't make the protein from Mimi virus. And so, well, there are lots of other giant viruses out there. So they found another one that had homologues of of these proteins, VIF4A, 4E, and 4G, in coton virus. Coton. You have to say it French, I guess, because there's only one T, coton virus. Coton virus japonicus. And they're 80% amino acid identity with MIMI. So they use those proteins in the biochemical assays. And in fact, they find that VIF4G interacts with each protein, 4A and 4E. And it also forms an intact 4F complex. So it's pretty interesting that even this short thing can associate with all these other proteins, because the eukaryotic one is pretty long. But anyway, this kind of indicates that this mode of interaction and formation of this complex is really conserved, right? So they have a structurally diverged or a sequence diverged 4F complex that interacts with the ribosome. All right, so big deal. It interacts. Does it do anything? That's the real question. So is it required for viral replication? What do? You knock out the gene from the virus, right? They use homologous recombination here, not CRISPR. Interesting, right? And they can make delta 4A, delta 4E, and delta 4G. The 4A and 4G, they could only make virus in amoeba that make the protein that's knocked out. So,
and those are engineered amoeba that they have
right.
introduced these viral proteins into. And by the way, we're talking about, even Matthias was talking about, sophisticated technology in these systems. This is new to me. I've never, of course, I am focused on this for quite a while, but I've never seen this kind of genetics with the giant viruses before. Making a complementing cell line and knocking the gene out, which is, critical to asking the question, are these just hanging out there, or are they really doing something?
So basically, the delta 4A and the delta 4 virus, you can't make particles unless you produce the protein. So they're obviously needed for particle formation. We're going to see why in a bit. Okay, so keep that in mind. But anyway, they can make viruses with these genes deleted, and they are really sick, right? They replicate 3 to 5 logs less than, in viral titers, less than wild type virus. And a log, folks, is 10-fold. So 3 is like 1,000, right? 5 is 100,000. 6 would be 1,000,000. Okay? Instead of saying, I was thinking the other day in the car, in the train, people say, why are you using scientific notation? Really? You want to write 1 with 9 zeros behind it all the time?
So true. And you're sitting there counting the zeros. Is this 1,000,000, 10,000,000? What is it?
And now scientific notation makes it easy to do math, right? Because if you want to subtract 10 to the 6 from 10 to the 9th, it's 10 to the 3. Just subtract the exponents. What could be easier? As someone who is weak in math, I love it.
I was talking to a well-known virologist. I'll tell you after who it was. And she said that she was at some kind of study section or scientific grouping or something. And they were talking, the virologists were talking about log units. And the physicists were clueless. They had never heard that terminology of log units. So, yeah.
Well, they have bigger units because their distances are huge. Yeah. All right. So anyway, these sick viruses, if you grow them in amoeba that make the deleted gene encoding the protein, then they're fine. So so it shows that they're essential for virus reproduction, but also the host orthologs cannot complement their function.
Right.
Right. That's really a cool thing, right? So the host EIF for A, E, and G, they're there, right? The host wouldn't grow without them, and the viruses will not grow. So they need them. It's really interesting.
So they need them, and they're doing something special.
Yeah, well, they're somehow interfering with making particles, but that could be anything in this cycle, right? So the conclusion they make is giant virus-encoded translation factors, well, these 3 anyway, these 3 are essential for viral replication and functionally distinct from endogenous host EIF4F homologs. All right, so now we do some experiments to find out exactly what's going on here. They say to define the molecular function. And Bernie Fields would say, it's just function. It's not a molecular function. He used to get very mad. So Rafi Ahmed trained with Bernie Fields first. He got his PhD with Bernie, and Bernie was very stickler about nomenclature. Used to bother him when people said, I'm going to study molecular pathogenesis. And he used to say, you're going to study the pathogenesis of molecules? Really? Okay. So they do ribosome profiling and ribosome RNA sequencing of amoeba infected with wild type virus or the deletion mutants at early and late time points. So it's 4 hours is an early and 8 hours is a late time point.
Basically, a way to identify the sequence of the viruses that are bound to the ribosomes under any particular condition.
Right. So what they find is that if you take away the, well, in this case, 4F, all 3 proteins, you get a decrease. You don't get a change in viral mRNA levels, so it's not needed for transcription or processing of mRNA, but you get a severe decrease in late viral translation, not early. Okay, early on, the translation of viral proteins, even in the absence of this VIF proteins, is okay, but late, it's way down. It's 50% decrease, the mRNAs that encode these are also expressed late. So they're not expressed early, these viral IFs. And so they're saying, well, there must be a switch between regular translation early, and then somehow these VIFs are made, and later on they get incorporated into the ribosome.
Vincent, do you have a sense for when DNA replication happens? Like when is early versus late for these this giant virus? Kind of.
I don't know the timing.
Yeah.
I don't know the timing.
Well, according to their figures, the time points they use are 4 and 8 hours.
Yeah.
So I'm going to say 6 hours. Is
yeah, that's right. That's probably right. That's probably right. Because you wouldn't make late mRNAs until you start DNA replication, right? So probably, if they're already being made by 8, 6 hours would be it.
Actually, that's a subtlety. I don't know if it even applies to these viruses, but in many DNA-containing viruses and phage, late transcription is coupled to DNA replication. So if you don't replicate DNA, you don't make late messenger RNAs. I don't know if that's true for these. I wouldn't be surprised.
Well, so there are late proteins defined in these virus-infected cells, right? And they're structural, typically structural proteins. They have the major capsid protein, the core protein, and some others. And they are highly translated at 8 hours compared to 4 hours. And without an intact VIF4F, these the mRNAs corresponding to these late proteins are reduced 2 to 14-fold on the polysomes. And so you're reducing the translation efficiency of these late proteins by removing this VIF4F. So there's a transcript-specific role of these factors in protein synthesis. It's pretty cool. Now, of course, if you take away the late structural proteins, you don't have assembly of virus particles. And so they see a major impact on virus assembly. So these viruses, like Rich's viruses, pox viruses, reproduce in viral factories in the cytoplasm. And they see viral factories in the cytoplasm in cells infected with these mutant viruses, but there's no virus particles coming out of them. Whereas wild type, you see lots of virus particle coming out. They call them pseudo-icosahedral nascent virus particles. Those are the wild type anyway.
Yeah, and then they have some malformed ones from the mutants.
The malformed ones from the mutants. So basically these viral translation factors, VIFs, provide regulation of protein synthesis, which in turn regulates virus assembly. So you have a switch later in infection to translation that's dependent on these VIF4F, and that's how you get the structural genes translated. And I presume that these, I don't know if they did it, I don't remember, but these VIFs don't translate cellular mRNAs, I'm pretty sure, right?
They do something that shows they're not interchangeable.
I'm
That's
gonna.
later.
I have.
Yeah. So, and according to the graphical abstract, it looks like host translation is down later on, if not shut off.
Okay. So I'm gonna complain about this a little bit. In their graphical abstract, they show the. Actually, I don't wait. Let me wait till we talk about caps and 5' ends.
Okay.
Also, if anybody wants to see in figure 3F, if you have the paper, I think, because I don't look at electron microscopy that much, but the transmission electron micrographs, you can see the actual viral particles. It's really cool because I don't normally look at these things. So I think that's really cool.
I'm glad you appreciate this because this makes me drool. Okay.
I do.
This is the kind of stuff we used to do all the time because in poxvirus infections and these giant virus infections, these sort of late effects in the factories and stuff are really obvious. And so this used to be a standard way of phenotyping aberrant infections of one sort or another. And they're really interesting.
So cool that you can actually see them like that.
Yeah.
I mean, anyway.
All right. So remember, the EIF4F complex recognizes the cap. The cap is basically a G residue in a reverse chemical linkage. It's 5' to 5' rather than 5 to 3'. And it's also methylated. And so this is a defining feature, this methylated cap of eukaryotic mRNAs. So how both the viral and the cellular mRNAs have this modification. So how would a protein distinguish between the two, right? So now they want to do some structure analysis to find that out.
And once again, for the lay listeners, messenger RNAs are these linear things, and they have polarity. We're talking about a 5' end and a 3' end. Just think of it as left and right, okay? And when they're translated into proteins, the ribosomes start at one end, the left end. And that, among other things, has a structure on it that Vincent just described, which is called the cap, which is a special nucleotide. And the ribosomes, when they initiate, part of the initiation process is recognition of that. And this factor is involved in that, right?
Yeah. So, yes, exactly. So the thing is 5' non-translated region. So you have a cap and you have 5' untranslated region. Then you have your initiating AUG. In eukaryotic mRNAs is not very conserved, but they say here the 5' UTR of viral RNAs has strong sequence conservation that's not observed in the host. So they say, man, maybe that's involved. Maybe the cap recognition is just more than the cap, right, for viral IF.
For these giant viruses.
In the figure, that difference between the host and the viral messages is really striking, okay, in terms of the sequence. And I will tell you, this is a bit of a digression, but in poxviruses, the late RNAs in particular have what we call a poly-A head, which is a whole string of A residues that are not encoded in the template. They're put there during the initiation of transcription by the polymerase kind of stuttering and with the effect that it adds a whole string of A residues and it gets capped and translated. And in poxviruses, which are, sort of the children of the giant viruses, they used to be the biggest around, but no longer. That poly-A head is significant in distinguishing viral RNAs from cellular RNAs. And this reminds me of that, put it that way. It's not just poly-A, it's either poly-A or poly-U or AU. It's AU rich, okay? But it made me wonder whether those are even templated or what their evolutionary relationship is to the poxvirus situation. But it does distinguish them very starkly from the cellular messages.
So they look at the sequence and they see there's a conserved A residue at the +1 position, which would be right next to the cap, right? And this is not present in eukaryotic cellular mRNAs.
So that's where I'm going to take issue with this, because the 2' O meth. I'm sorry, Vincent. Am I jumping the gun? Did you methylate?
Yeah,
that
I didn't get the methylation.
Okay,
yet.
sorry.
Yeah. That's okay. So they look at binding of VIF4E to nucleotides that mimic the structure of CAP. So the human EIF4E binds to anything that has a 5' M7G CAP, okay? But it doesn't interact with GTP because it doesn't have a CAP structure. But VIF4E interacts with a viral 5' cap M7G if it has a 2' O methylated adenosine next to it. And it binds really badly to M7 GTP or anything without an unmethylated +1 nucleotide. So they think this is giving specificity of the VIF or F2 viral messages.
So this is where I have the issue, because in their graphical abstract, they show the cellular messenger RNAs as not having the 2' O methyl adenosine, and that's not true. Cellular messages have at least 30 to 50% of their messages have a 2' O methyl adenosine, and maybe even more. So there may be some discrimination based on binding of that methyl group.
So you're talking about in the +1 position?
Yeah.
Not just throughout the message, right?
No,
in a
+1 position. So
okay.
there's it's nearly 100% of messages that have either a 2' O methyl adenosine or a 2' O methyl uracil or whatever that is. Okay. So most of the now I don't know about amoeba, but dictyostelium does. The position 1 has a 2' O methylation in almost 100% of the messages. Okay. So this aspect of the difference, I think, let's put it this way. If I was reviewer 3, I would ask for a discussion of the abundance of those modifications in the cellular RNAs, because that's not made clear in the paper.
Yeah.
So that makes me think
for eukaryotic messages, all you need is the M7G to bind the EIF4G. You don't need anything else. Right?
Yes, that's true.
But it begs the question why dictyostelium has the 2' O methyl A.
Well, I'll tell you this. It is to a large extent in eukaryotes a molecular marker of self. Okay.
Yes, it's true. It's
because it marks them as eukaryotic messenger RNAs. Because in fact, poxviruses, their capping enzyme just puts on the M7G. They have an extra enzyme that does the 2' O methylation. And if you knock that sucker out, the poxviruses lose virulence in vivo. It still grow fine in cell culture, but they lose virulence. Actually not presumably, because the cells recognize the unmethylated RNAs as foreign, and that triggers a cellular immune response.
Right.
Okay.
So look at figure 4A. It's a distribution of bases at the +1, actually the first 10 bases. But look at the +1 position. The top panel is the amoeba. All 4 bases are found in equal numbers at the +1 position. But if you go down to the virus, it's almost all A and a little bit of U.
Yeah. So if only if I'm right that only 2 of those residues are really subject to the 2' O methylation, that would suggest that there may be as much as 50% of the cellular messages that are not methylated at that +1 position.
Yeah. Right. Well, I tell you, then they're going to go on to do the structure of the crystal structure of VIF4E together with M7 with a methylated A next to it, okay, and a U, right? And you can see the structure shows there are conserved features with EIF4E, and then there are novel features that make the viral protein, bind preferentially to viral RNA because of that methylated A there in particular. The way, and I won't go into it in detail, but there are differences between the way the protein interacts with the M7G in human EIF4E versus the virus. So for example, in the human, it binds M7G in a concave binding pocket. Okay. In 4E, the M7 is recognized in a similar pocket, but it's not recognized as well. It's not bound as well. The geometry is slightly different. And that, they say, explains why it's a weak binder of M7G. But then there are residues that specifically coordinate the 2' methyl A and make for a stable interaction. There are 3 amino acids that are conserved. And if they change one of them, it disrupts the binding for the cap with the plus 1A. So they say that the region of 4E is not positioned to make such contacts with the A nucleotide if an A nucleotide was there. So, Rich, to your point, yes, a fraction of the eukaryotic messages may have methylated A at that position, but EIF4E can't bind to it because its structure is different from VIF4E. Right?
Right. Okay.
You understand what I'm saying?
Yes. And that's clear from their binding studies, that the host cap is not recognized by this factor. Okay, fine.
Yeah. And so they do biochemistry with altered proteins to confirm this, that VIF4E has novel adaptations to recognize both M7G weakly and M7G with 2' OMEA. Okay, so it will still bind M7G, but the addition of 2' O methyl makes it a really good binder. Whereas if the RNA doesn't have 2' methyl A at the plus 1 position, it's going to bind weakly. So they do, they validate these in cell lines. So they engineer the amoeba to have these changes that would disrupt the 4A binding, and they don't support replication of the virus. So basically, they say they couple canonical M7G binding with a novel mode of modification-dependent recognition, sequence, which is the A, and the modification-dependent, which is the methylation, to get preferential. I mean, it's preferential binding. It's not absolute, right? It's preferential binding to the viral mRNAs. Anyway, so I think that's a really cool.
They mention this. And it will require ultimately further study, but I will bet you that AU-rich leader sequence downstream plays a role in all of this as well. Yeah,
that's striking. It's all it's all AU, right. And you look at the host, and it's just heterogeneous, right? It's all 4 bases. It's very interesting. Okay, so then they do some experiments. So they want to, what kind of selection would drive the evolution of this in a virus, right? And they say, well, these giant viruses infect a lot of different kinds of protists that undergo stressful environmental conditions, right? They just Siberian permafrost, pH differences, temperature differences. So they're wondering if these viral EIF4Fs provide relief from these stress conditions. Okay, so they infect cells with virus, and then they subject the infected cells to a bunch of conditions that may reflect environmental stresses, and they find that the wild-type virus is resistant to nutrient deprivation, chemically induced ER stress, and oxidative stress, and if they take away the viral 4F, it's even worse reproducing under those conditions compared with baseline. And they also look at another virus that doesn't, anemia virus, which doesn't encode any VIF4, and it's trashed by these different conditions. So they think maybe this VIF4 gives the virus the ability to replicate under stressful conditions.
And I would say that if you're a cell, there's hardly anything more stressful than being infected, especially by a giant virus.
Yes. So that's basically it. That's the story. They have a novel translation protein complex. It's turned on late, which preferentially translates the viral late mRNAs, and that makes capsid proteins so that you can make virus particles. And maybe it also confers resistance to stressful environmental conditions. So this is really the first example of how such a viral translation factor would be useful for the virus, right?
I think historically there have been these things recognized in some of these viruses, but
Yes.
but this is the first example of really nailing it down and saying, yeah, they're required. And it's not just the factors; the messages are different too. Make a
Yes.
a special kind of message and a special kind of factor.
Yeah, it's
and
quite
now you have
interesting,
a something that's very virus specific in the back half of the infection. It's very cool.
Yeah, they say we solved the long-standing enigma of why giant viruses encode large complements of cellular genes that are absent from the rest of the virome. So, I mean, I'm sure there's a lot more, right? These other viruses may encode other parts of the translation apparatus with other mechanisms. So this is a really interesting mechanism that it's recognizing this methylated A, right? But there probably are others too.
Well, and among their limitations of the study, they say increasing evidence indicates that translation during APMV infection is spatially organized with the viral mRNAs, tRNAs, and translation factors co-localizing in the zone immediately surrounding the virus factory. This has always interested me, and it goes back to Angela's comment about the electron micrographs. The virus establishes a very distinct compartment in the cytoplasm, and that may involve localization of all of this activity. So there may actually be a spatial organization in the cell that contributes to this distinction between viral and cellular activities. Mm-hmm. Very cool. Matter of fact, I mean, I'm sitting there reading that bit, and Angela, there must be some of these experiments out there. But immunofluorescence with antibodies to these factors or immunogold staining of sections ought to show you that spatial organization of these factors. That would be fascinating.
That would be beautiful.
So, by the way, they mentioned in the limitations, we note that work on giant viruses and non-model organisms like acanthamoeba is limited by the lack of well-annotated genomes and transcriptomes. For example, the relatively small number of annotated host 5' UTRs limits our ability to determine how the identity of the +1 nucleotide impacts translation of host mRNA.
Okay. So they may not even, for amoeba, they may not even have the data on what the fraction of 2' methylation is in the messages.
Or not much data.
Or not much data, yeah.
But anyway, if you're wondering why this is important to do this gives you insight into evolution and how things evolved, the translation system, how it evolved, right? And so if this particular system evolved as in a response to environmental stresses, then maybe it also turned out to have impact in separating viral and cellular translation. And you know what? Who knows what it will be useful for, but it might be. You just never know.
Well, from this scientist, I'll tell you, the importance of doing this is that it's really fun. Okay. Okay?
It is fun.
That's what's motivated me the whole time. It's really fun.
There are people now in charge who don't care if we have fun or not, okay?
You're not allowed to have fun.
They say this, and so we have to come up with other reasons, and there are plenty. Anyway,
I can see myself. I can see myself sitting, looking in the microscope. Okay, I spent hours doing this, and if you with an electron microscope, you're sitting at this table, looking through a window at the sample. Okay, and you got these knobs that move the sample around. Okay, so you're looking through a window. It's almost like you're in a spaceship looking at the surface of like the moon or something, except it's the interior of a cell. And when you twist the knobs, it's like you're driving around over the surface of this thing looking for stuff. Same goes with an immunofluorescence experiment. It's just, and I'd sit there and I'd think, nobody else in the world has seen this. This is incredible. Great stuff.
I think you should have fun at what you do.
Absolutely.
Right? Even if you're a surgeon, you should enjoy it.
I'll bet you they do too. Yeah.
Well, to me, I can't imagine enjoying getting up at 5 a.m. and going and cutting someone open. I just can't handle that responsibility, but I think they enjoy it. And people who have less interesting jobs, they don't enjoy it, and that's sad because they're not as fulfilled.
So
But
to
that's
Kathy's point, where I feel like a lot of people, considering the title of this paper, as Kathy said, the name of the paper, a lot of people I think will expect us to at least have a small conversation on maybe reductive evolution, or if this could be some sort of, life or was life. I know that you guys still are in the know. I'm guessing you're in the viruses aren't alive, but the title of this paper lends to that, to talk about, is this something that is now like devolving? Are these viruses, were they once some sort of organism and this is like vestigial, like remnants of it? Or what do you think? What do you all think? Because I'm not a virologist, so I like to ask these questions.
I think it can go both ways, right? It can be both. Why not?
In the first several dozen episodes, so shortly after I joined TWiV, we would have this conversation almost every episode, whether or not viruses are alive. And we actually took a poll of the listeners at one point. I think it came out about 50-50. And one of the things you can say is the virus particle is not alive, but an infected cell
right
is living. These
But
in a
that's
way,
the
these are
cell.
kind of crazy distinctions. This goes, I think we talked about this last time. We try and draw these black and white boundaries in our descriptions of science, and it's not that way. It's gray.
But are giant viruses remnants of cells? Sure, it could be, but also cells came from viruses as well. I think it can go, because we had an RNA world,
right. Can go
and then
both ways.
the cell world came after it. So I think both are compatible.
They're probably co-evolving
Yeah.
all the time and swapping genes back and forth. I mean, DNA is really a problem, right, Vincent? It's everywhere.
It's everywhere. Hey, let's do some.
I was just going to say,
emails.
For me,
Yeah.
They could have just left off the of eukaryotic life, like leaving out hallmark. So giant DNA viruses encode a translation initiation complex. That would have been fine. But they did kind of stir up this idea of, well, is this, getting us closer to a living virus? And as I said, I think it's what the popular press picked up on.
Well, exactly. And that's why they put that in the title, because then we're going to talk about it. And that's why the presser is going to talk about it, which maybe is good, because then they're going to look into the paper. And if they hadn't titled it like that, then people wouldn't be talking about it.
I'll give them a break and say the reason they put that in the title is because they were having fun.
Also,
it's okay. Yeah. By the way, this idea of having a late translation factor switch the program from cellular to viral. Don't some phages encode transcription factors that are made later and make late genes, transcribe late genes, right?
Yeah, that sort of transcriptional control has been going on since day one. And people have been suspicious of translational control, but that happens too, that once again in pox viruses, and Kathy highlighted some of this stuff, there are tRNAs and that kind of stuff, but there's been within the last few years, a paper on pox viruses from Derek Walsh's lab that shows that late during infection, the virus makes I don't know all the details here, makes a kinase that phosphorylates a protein on the ribosomes and alters the specificity to tilt the translation towards the viral messenger RNAs, which, as I said, have these poly-A heads on them. Okay.
Right. Let's do a couple of emails. I'll take this first one from Pseudonym. He or she sent a cartoon, and it's three people in a lab. Two of them are straight-laced guys with ties and lab coats, and the other is a young guy with fuzzy hair and a different lab coat, right? And the guy with the fuzzy hair is saying, "These days, if you're not a mad scientist, you're not paying attention." Avid listener since the pandemic, stayed for the science. Love to the whole crew.
Great.
Thank you.
Thanks, Pseudonym. It's a good comic.
Yeah. Rich, can you take the next one?
Bill writes, TWIM 1299. It must be TWiV. 1299 comments.
Oh, sorry, TWiV, yeah.
2 follow-ups that might be of interest. 1, The fall armyworm, Spodoptera, I have trouble with this.
Frugiperida.
What was that?
Frugiperida.
Frugiperida. Is near and dear to people who express and purify recombinant proteins, as cells from the ovaries led to SF cell lines used in the baculovirus insect cell expression system. Cool. 2, from Rich's Pick of the Week, listeners might want to check out Ken Burns' documentary, his first, 1981, Brooklyn Bridge, that was narrated by David McCullough, as were several of Ken's documentaries. Yeah, Burns, McCullough is just a delightful narrator. He's got this voice you can't not listen to. Burns' Brooklyn Bridge was nominated for an Academy Award and inducted into the National Film Registry in 2025. Good point. I think I've seen that, but I should look it up again. 3, -1C and partially sunny in Frederick, Maryland.
Mm-hmm.
Bill. Excellent, Bill. Thank you.
Kathy.
John writes, Doctor's TWiV. Fascinating dive into the basis of clotting with the adenovirus vaccines. Thanks. And with the moths, someone in my old department in the botany division worked on cabbage moths. Not sure if those were the same species you talked about, but what I particularly recall was that male cabbage moths expend 13% of their body mass in mating. Choose your mate wisely, I guess. But with the picks, I first thought that I'd just write this to Rich. Against impossible odds, you picked the very book I've just about finished. The story of the Brooklyn Bridge is truly astonishing, but as far as the book itself, it's a real example of how a consummate writer like David McCullough will keep writing to describe something rather than just add a figure. For the most part, I gave up trying to follow his descriptions. John McPhee, Annals of the Former World, is another example. Fortunately, the scientific literature, including patent applications, isn't like that. But then, Alan, if you like Southern cuisine, you must like collards. There's a great book on them that you aren't likely aware of. Collards, A Southern Tradition from Seed to Table. And he gives a link for that. Ed Davis is in environmental studies at Emory. We have a weird thing with ampersands coming in. At Emory University down in southwestern Virginia. Emory Henry U. Anyway, it basically covers the ethnobotany, sociobiology of collards. They traveled around the South talking to people with their own collard patches, and they go into considerable depth asking the question where this humble variant of Brassica oleracea originated from in the first place. Collards first resonated with me after having them in a cream sauce made by the mother of a fellow grad student in Ashland, Virginia, north of Richmond. There was no shortage of butter in the sauce, but she lived to 97, so they must be healthy. This was over 50 years ago, too, so they must have been good if I still fondly remember them. And Vincent, thank you. These people who think we're going to colonize Mars are just plain loony. Less than 10 years ago, I listened to the director of the Space Center in Huntsville speaking about that. She said that either Jules Verne or H.G. Wells, if I recall, estimated that 90 advanced trips would be required before a manned trip, and that was about right. But then she continued as though they were seriously planning for it. I wasn't convinced. Otherwise, the best that I can say is that February is over 90% over. Regards, John.
Angela.
Gretchen writes, I saw this poll in Reuters, and the poll is named
Reuters.
Reuters. That's such a weird way to pronounce how it's spelled. Anyway,
well, this is because you're not British.
It's true. So it's called Americans Trust Vaccines and School Mandates. So they saw this poll in Reuters, and was more hopeful about common sense and perhaps all the good pushback coming from sensible people around the importance of vaccines. Just thought it was a balanced view to acknowledge. Thank you for continuing to report on science. Gretchen Bingham Toronto, or Toronto, as we would say it. But so it looks like it's on vaccines. It's called Americans Trust Vaccines, School Mandates, Rejecting Trump Agenda, Reuters LP SOS poll finds.
It's interesting. They're polled according to all adults, Republicans and Democrats, right?
Yeah.
Mm-hmm.
And it's politically divided. right? Like, should the government require children to be vaccinated? Republicans, 33% say no. Democrats, 8% say no. I mean, it doesn't make any sense, right? Except that it's a political issue.
But it is, I think, striking that the majority, regardless of political party, the majority think that kids should be vaccinated to go to school, and that the vaccines are safe. I just saw an article this morning that maybe it was even HCR saying that they're kind of almost backing away, not making too much noise about RFK recently in the government, because they're starting to realize that maybe his anti-vax thing is appeals to only a minority of individuals. That
But
would
also
be
it's
nice.
slightly worrisome that in the Republican on this poll where it says should children have to be vaccinated to attend school, 30% said no, which is slightly horrifying. But anyway, that's higher than I would think.
They asked them, do you have a generally favorable view of RFK Jr.? Republicans, 72% said yes.
Yeah.
That's crazy.
Democrats, 12% say yes. That's not based on any knowledge of what he says, okay?
Right.
If you're saying it's ridiculous.
No.
Should the government recommend fewer vaccines for children? Republicans, 47% yes. Democrats, 13% yes. We always have known that these questions are politically divisive, and it should not be, right? It should be based on science.
That divisiveness became much more pronounced during a pandemic.
Yes,
because of
and after,
whether or not you should mandate certain practices like vaccination or masks or whatever. And this is where people really kind of push back.
Let's do a couple more.
Before you go on,
I
wasn't sure, but I checked. Reuters is derived from a German surname. It was the news agency was founded by someone who was born in 1816 in Kassel, Germany, Paul Julius Reuter.
And is it supposed to be pronounced Reuters? Does it
Reuter.
say?
Yeah, Reuter is the German
Okay.
pronunciation. Yeah.
German pronunciation, correct. Okay.
I'm sorry, Angela. I just had to correct you.
No. I think it's great. I'm like, maybe in German they say like Reuters, or I don't know, something else that's like.
Yeah, I was wondering if the EU is not substituting for an umlaut somewhere, but I don't think so.
No, I don't think so.
Anthony writes, when I was a child in the 60s, fishing and crabbing in the Hudson River on the Jersey City side across from Manhattan was very popular. Nobody who did this had any economic issues that prevented them from shopping at a grocery store. The river was so polluted then that there was one time that retching prevented us from approaching.
God.
What we did was go to another spot a bit upwind. Nobody gave a thought to the possibility that the crabs and eels were unsafe for human consumption. In the 80s, an acquaintance was doing initial work for a construction project at the outlet of a New York City sewage treatment plant. He saw an elderly individual with many drop lines and bins of ice with striped bass. It seems that the effluent attracted small marine life, which in turn brought in striped bass. The worker asked the local fisherman what he was doing with all the fish. The answer was that he was selling them to the finest restaurants in Manhattan. Fresh caught indeed. A few years ago, someone from Zimbabwe showed me photos of his family's home. It was finer than any property where I ever lived. He told how his father would bring home roadkill to cook. Africa's a big place. If people consume bush meat at all, and they reason why they do, might change in just a fraction of a mile. I like that.
Anthony.
That's a good point. Mm-hmm. Very good, Anthony. All right, I want to do the next couple because, there are only two more. Back to Kathy. Who did the first one? I don't even remember. I did.
I think it was
Did Rich do any?
It was Rich. He did.
I did one. I did Bill.
Mm-hmm.
You did the cartoon, Vincent. So,
okay, you do the next one, please.
Where
Maria.
are we? Maria writes, Dear TWiV, not sure if this will make it in time for February episode, but I wanted to share this story that was new to me. My son is in second grade and was assigned Wilma Rudolph as a notable African-American person to research. What an inspiring person. She gives a wiki link to Wilma Rudolph. Most relevant to TWiV, she had polio at the age of 5, which left her wearing a brace. Through medical attention and a very supportive family, my second grader's book estimates more than 10,000 massages given by her siblings at home. She not only managed to stop wearing the brace at age of 12, but became an athlete and won a bronze medal at the Melbourne Olympics at age 16 and 3 gold medals in the Rome Olympics at age 20. Holy cow. She was recognized as the fastest woman in the world, and upon her return from Rome, required that her celebratory parade was integrated. And it became the first integrated event in Clarksville, Tennessee, her hometown. I've got chills here, dudes. Mm-hmm. Wow. I know it's a tenuous link with TWiV, but I thought you may appreciate the polio connection. Absolutely.
Definitely.
Thank you so much for everything you do. I deeply value your candor, depth of knowledge, and perspective, as well as the fact that you share them with the world for free. I may not always agree with everything you say. I'm an economist, but I don't agree with everything anyone says. At least I trust that you don't have ulterior motives. Writing you with the very best, Maria. She makes me think my son-in-law is an economist. And every time I ask him an economics question, the answer is prefaced with, it depends.
Well, that's kind of like a scientist answer too, no? Yeah. Also, it depends.
Oh, did you see the Surgeon General confirmation hearing? Every time they asked her a question, she said, "Oh, it's complex."
Yeah.
Do you approve hepatitis B birth dose? Oh, it's complex.
Yeah. No.
No, it's not complex.
No,
it's not complex.
Not really complex.
Bullshit. All right, one more. Kathy.
Ken writes, thank you for the fine science-based education you and your esteemed guests have provided me since I discovered TWiV in the early days of COVID. I live in a rural area of Northern California, and 70% of my fellow county residents are right to far right leaning politically. I'm an exception. I know from experience that I cannot convince these people that their trust in a particular political leader is misplaced. However, I do believe many could be convinced that particular positions are wrong, as long as I have credible facts. While it feels good to hear criticism of the CDC or FDA in its current iteration, I know such commentary would shut down further consideration of any studies, arguments, or any other fact-based positions I might want to offer these folks. I truly believe a politically non-critical broadcast could bring in more viewers, listeners, and thereby expanding knowledge beyond those who are already believers in science. I should note that my bride of 40 years thinks they wouldn't listen anyway, and you should let her rip. But then doesn't that leave you preaching to the choir? Perhaps there's room for a politics-free version of your show. Change can only be made in increments when the public is so far apart. I love what you do and will be a forever listener. Happy trails, Ken. P.S. I listened to a stock brokerage podcast from a major brokerage company and have no idea what the political position is of the host.
Well, of course. Otherwise, you're not going to, if they're not agreeing with you, then you're not going to buy from them or whatever.
Right.
I don't think we would get more people. I really don't. I think science is a problem for most people. That's it. Unless you're lying about the science, because there are some people who lie about science and they have 3 million followers on YouTube. And we give you the truth, and we have 141,000, okay? So, and it's not because of politics, because we never used to talk about politics. And anyway, we don't talk about raw politics. We talk about it as like in Democrats voting that you could keep the vaccines as they are and Republicans not. That's absurd, right? It's mixing politics with science. We have to talk about that. Mm-hmm. Anyway.
You said 141,000 listeners.
On YouTube.
On YouTube. Wow.
Only YouTube. The audio is different. It's a different story. But
yeah, but still. I mean, so it's even more than 141,000 total, right?
Yes, but it's not 3 million.
No, but that's okay.
No, it's okay. I'm okay with it. I don't think we're preaching to the choir. Well, you know what? We mostly are preaching to the choir. I think most people who listen agree with our position.
But this way, the choir is learning the music and may be able to spread it to others who haven't heard the music yet.
Yeah, I agree.
That's cute.
So I was thinking about this as we were reading these emails. Many of these people say they joined during a pandemic, and they're still here. That's
Mhm.
good.
That's good. Yeah, it's great.
Mhm.
No, I get emails every week about, I can't handle your show. There's too much politics. I'm not listening anymore.
All right.
But that's just what's
I mean-
happening. We have to discuss it. We can't just blindly ignore everything that's happening.
But
especially
90% of our content is
not
pure
that.
science.
Of course,
Mhm.
of course.
Right? It's just that these people don't like our position. It's not that they don't like politics. They don't like our position.
Mhm.
And I would rather you said that. They say, TWiV, I totally disagree with your political position. Fine, you have the right to do that. But don't tell me not to talk about it. That's absurd.
Mhm.
Let's do some picks. That's more fun. Angela, what do you have for us?
So my pick this week is actually a book that came out in 2022. A lot of people have heard of it, but for those of you that haven't, it is called The Song of the Cell by Siddhartha Mukherjee. I think that's how you say his name. Hopefully.
Reuters. It's pronounced Reuters.
I don't know if the H is silent. Mukherjee or Mukherjee.
Mukherjee.
Mukherjee. Is it Mukherjee? Okay. Are we sure?
think so.
Or are we also just saying that?
I think
Yeah,
so.
Mukherjee.
Mukherjee. Okay. Siddhartha Mukherjee. Also, he wrote the book, The Emperor of All Maladies,
Yeah,
I believe
yeah.
it's called. But The
Yeah.
Song of the Cell is so beautiful because I think for scientists it's beautiful and for non-scientists. So for non-scientists, it has like this beautiful, he has a beautiful way of writing, explaining, every day, every second, your cells are making life and death decisions throughout your body. Immune cells are patrolling around. He's kind of anthropomorphizing, but in a very beautiful way, how our bodies work and explaining at a wonderful level. He goes into details, but not so many details that it loses the listener if it's someone that's a non-scientist, but also just highlights the complexity of our bodies and about everything that's going on, quote unquote, automatically. And I think that it's just a very awe-inspiring book for people that aren't scientists and even people that are, as I'm reading it, because I'm still in the process of reading it. I think it's so beautiful how he's explaining the body and also thinking of cell types that I normally don't think about, like neurons, and how he has like this nice way of saying, well, to explain depression, that the neurons aren't communicating together because they're missing signals between each other. And I think it helps a lot. It would help a lot of people understand certain diseases better and just understanding themselves, because all of this is billions of interactions that are going on inside of our bodies at every single millisecond of the day that we don't appreciate. And this book will help us appreciate it a little bit more. So
What's the key of the Song of the Cell, Angela?
what's the
B
key?
flat.
What do you?
It's in B flat.
I don't know. Rich would know. B flat.
B flat.
That's
Actually,
not
I just-
is that a minor key, B flat?
It can be. Depends on.
You have to say B flat major or B flat minor.
Yeah.
Okay.
The key of D is supposedly a happy key, and it was used a lot for weddings or music for weddings or Easter or something. I don't know. I just was at a concert and they talked about that. Different keys have different things associated with them.
Yeah. Somehow you probably are more sensitive to this than I am, Kathy. Somehow barbershop harmony, the predominant keys are B flat, A flat, and E flat. And I don't know why. And just recently I was with one of the quartets I sing with, singing a song, Ebb Tide, actually, which is a stretch for the tenor who has the melody in for most of the song. And he said, let's try this down a half step. Okay. And so we tried singing it down a half step, and it was a train wreck. I could hardly recognize the song. I could hardly sing it. It was really weird. It's just a half step, right?
Mm-hmm. Mm-hmm.
It's the most exaggerated example I've seen of that kind of thing. Really
That's
weird.
interesting. Yeah.
The Righteous Brothers.
The Righteous Brothers.
Kathy,
Well,
what do you have for us?
this is what I made the allusion to earlier, that should almost be a snippet because there's a lot of science here. Anyway, my older brother sent me an article from the Wall Street Journal, which I think is going to be paywalled for everybody. So I found some ways that you can look at it. But the title of the Wall Street Journal article is, "It's called the Fitbit for farts, and it's no joke." Okay, so this guy's article came out February 20th. But the University of Maryland put out a press release because that's where the science comes from. And that was February 10th. And I can't imagine where the press has been ever since February 10th. They totally missed this. But so the deal is that these scientists have been working on a way to monitor the gut microbiome by measuring, it's called flatus hydrogen. So flatus is another word for farts. Okay.
Flatus is great.
Flatus. Okay. So, and they published a primary article in Biosensors and Bioelectronics X. But they're working on the gist of this, most of the stuff that's in this Wall Street Journal article and some of the stuff in the press release is newer stuff. And I think this Wall Street Journal guy went and he's put in a picture of the sensor, a graph of flatusomics. It's a flatusomics dashboard. The gist of it is that they've made this sensor that can measure hydrogen, which is what the gut microbial activity produces. And in fact, it's only the gut microbiome that produces this flatus hydrogen. And so they designed something called smart underwear that people could wear comfortably up to more than 11 hours a day with high adherence to the protocol. They could validate detection of different diet-driven shifts in the gut microbial activity with 95% sensitivity. The smart underwear allows longitudinal studies of gut microbial metabolism. So they use 3D printers to make these casings, which end up looking a lot about like an AirTag to me. They have a collection of mannequin buttocks in the picture that they attach to gas canisters to emit the synthetic flatuses. And also they use these to calibrate all their newly manufactured ones. They've made 800 so far. And because they've sent them out because they want people to wear these and get them data. And they've had 4,000 people write in already that want to be in the study. So they're frantically making more. And they have to calibrate them. So they do that with these mannequin buttocks. But here's the cool thing. Farts emit 20% hydrogen. And this guy can't have this in his lab because they're flammable. So he tests them at a lower concentration for a longer period of time. Okay, this guy's name is Brantley Hall. He's an assistant professor in the Department of Cell Biology and Molecular Genetics. Okay, so in the current study, people are taking a picture of everything they eat and drink, and then the top 3% gas passers will get a chance to have their microbiome sequenced. And that could lead to information about what microbes maybe are correlating or contributing to the excess gas. And so the picture in the primary article shows their first generation sensor that seems a little bit bigger than a quarter. Anyway, I think, so the new estimates are that there's 32 flatuses per day per person, the range being from 4 to 59. So this is more, also doubling the previous estimate, which is only 14 per day. And they can, what's good about this is that you can monitor it overnight. They don't have to depend on people's memory. And let's see. Oh, yeah. And they tested this with inulin. So I don't know if you've ever heard of inulin, but it's something that's found in Jerusalem artichokes, which when I encountered them, looked it up and found, yes, it's known to produce huge amounts of gas. So they're using inulin, which is a kind of sugar, to generate their stuff in the lab. Anyway, wow. The press release has stuff in it. I'm also putting in a link to the Wall Street Journal if you know of somebody who gets it or you want to go to the library and try and find it, because it's really well written and hilarious. So.
I guess beans would also increase flatulence also.
Which one?
Flatus.
Oh, beans.
Flatus.
Flatus. Yeah,
the word
is flatus. Yeah.
Well, it is flatulence. It's the same.
There's no difference.
Flatulence.
It's just the way to talk about it.
I just think it's hilarious that they called it flatus instead of flatulence or something else. They just new word, flatus.
Right.
I want to
well-
know if there's a difference between males and females in the frequency.
all
Or in the
kinds of
amount.
questions. All kinds of questions that could be asked now that they have these monitors. I'm sure we'll
And
have
that's
more
also-
data.
People want to wear it. What if they're on like the high end of like 58, 59, as opposed to, I don't know what people be like.
Oh yeah,
This is going to
there-
break up marriages.
yeah.
Or you change your underwear every day, but I guess the sensor just
Oh, I
comes
think you take
out.
the sensor out. Yeah, it's
And
So you're
probably
talking
a
about-
Velcro thing or something.
Is
During
it
courtship,
washed?
Oh, yeah.
you have to.
Yeah.
During courtship, you have to wear one of these things.
Yeah,
In
in
the-
the record holder from the published paper was 175 times in a single day.
Oh my God.
That's like every
It's
few
like-
minutes.
Oh,
Yeah. So,
That's
Kathy,
crazy.
this is brilliant.
well,
Thank you
and
so much.
That's
This
hilarious.
is
where
this
did you
is-
get this? Where
I
how
told
did
you
find
my
it?
brother sent me the article from
Oh,
the
your
Wall
brother
Street
sent it to
Journal,
you.
and
Okay.
then I followed that up, and then I wanted to find something that people could link to or could look at, and that's this press release from University of Maryland. Now, isn't that where Rich, the guy who had the cone thing that you would breathe
the
into?
Gesundheit 2.
Yeah.
Yeah,
I think
yeah.
he was
I
from
don't
the
know.
University of Maryland, but somewhere around there. Anyway,
I think it was. Yeah, University of Maryland. Yeah.
so so we will probably hear more about this. I can't believe it wasn't already in the New York Times. Science Tuesday, Evening News, whatever. But
Also, imagine like your lab. People ask you, "Oh, what do you study?" You're like, "Farts." Oh no, you say flatus to make it sound
Flatus.
better. But you
Flatus.
say microbiome effects.
yeah,
Flatus.
yeah. But you can be sure that the title of the article in the Wall Street Journal got a lot of hits with
Of course.
Fitbit for farts.
That's
but that's in
hilarious.
fact
Good.
what they call
I
it.
like that very much. That's great.
Excellent.
Rich, what do you have for us?
This is my wife found this in I guess yesterday's paper or something. Yesterday's Times. This is shades of various symbioses that we've been talking about. It's a Times article called. I guess the section in the paper is called trilobites. I guess that's a pun. It's called The Password That Lets Caterpillars Hide in an Ant's Lair. And the general topic is, let me see if I can find this word here. I forget. There is this general phenomenon that numerous creatures hack ant colonies
Mm-hmm.
in order to use them to provide protection, nutrition, and etc., including insects that hatch larvae that trick the ants into taking them into their colony and nurturing them until they hatch and come out and become adults. I just think that apparently this phenomenon has been known for decades or longer. And I vaguely remembered something about this. And mostly I just wanted to bring that up and read the article and look at the pictures because they're cool. I got ants taking care of a larva. But the paper that they cite and describe is also interesting because the signals that the larvae give off to trick the ants are numerous. There's chemical signals and et cetera. But in this particular case, what they're studying is an audio signal. The larvae apparently make sounds that the ants dig. And I think they dig them because they mimic to some extent. It was hard for me to understand. There's a lot of language in the paper. I got the sense
It's a lot of
that
words there.
that the sounds are familiar to the ants, as if the larvae have learned to speak ant. All right,
That's crazy.
and
That's
so
cool.
actually, the Times article describes it as a crying baby on the doorstep, which I think is a wonderful metaphor. And the ants take this thing in. And one of the things that I found entertaining about the paper is, as you might imagine, it got the really small microphones that they use to record these sounds, small and sensitive. So at any rate, have a look. It's very cool.
So this is interesting because ants are blind, right? So they depend on smell and apparently auditory cues. We had a guy on TWiV years ago, and he was showing us pictures. You know, when the ants are heading back to the colony, you got all these outsiders just in the single file, right? They're just trying to sneak in. And the ants can't see them, but they can sense them in other ways.
And auditory, I think, is I don't know if they're actually making sounds. They're somehow emitting vibrations.
Maybe vibrations, yeah.
Mm-hmm.
Yeah. That's so cool. But the lead author is Kiara.
Oh, there you go.
Look at that. All right, my pick is a book. I've done like 5 books in a row now.
Astrophysics.
Yeah, I'm into astrophysics now. This one is by Neil deGrasse Tyson. It's called Astrophysics for People in a Hurry. I'm not really in a hurry, but it's an interesting title. And it's just it's kind of a popular introduction. I really wanted to see how he would introduce a pretty complicated field, right? You know, all about astrophysics, or structure, the universe, its origin, how gravity works, light, dark matter, and energy. But he's a great writer. He's got a really good style, and it's funny, and it reads really quickly. So big seller. So this is great that he's reaching so many people. We were talking about this the other night. People love the space. They love it, right? Mm-hmm. And I love at the end of each of his shows, he says, "Keep looking up." I love that. It's like another TWiV is viral, right?
It's true.
Anyway, Astrophysics for People in a Hurry. We have a listener pick from Larry. This is really cool. Thank you, Larry. Are T cells early risers? All right, so it's an article, commentary by Derek Lowe in Science. And basically, there's some evidence that immunotherapy works better if you give the infusions early in the day rather than later. So they did a trial, and they give people who are getting infusion for lung cancer in the morning or later. And they got this huge difference. The survival rate was 28 months in the morning and 16 months in the evening for the two groups. That's a big, and he says, you don't even need statistics to know that that's important. And anyway, there's also an article in some journal, Nature.
Some journal. Random journal.
which is all about the trial. So if you want to read the actual data, that is so cool. And so Derek is like, what's going on? You know, there's some evidence that T cells are more active in the morning, and maybe that's part of it as well. So that's really funny. It's great. Check it out.
Yeah, there's quite a few papers now on even like neutrophils, on their function and how they have the circadian like spikes in neutrophils in your bloodstream and lower. There's actually quite a bit of research on that now, which is really interesting. It also makes you think about, when do you infect mice? Does the timing of the day matter? Like in our case, for example, or with humans in this case.
So here in this study, early was 7:30 to 2 p.m. So it's
pretty big.
Horribly early.
That's pretty big.
Yeah, it's a big thing. And then the other was 3 p.m. to 8 p.m. So I don't know, maybe you should get your vaccines in the morning, right?
Also, most people do, don't they? But that just might be.
I remember waiting for COVID vaccines in the dark.
No. I mean, at that point, COVID is a specific case.
Yes. All right, that's TWiV 1301 or 1301, 1301. There are a lot of ways you can say numbers. It's great. You can find the show notes at microbe.tv/TWiV. You can send us questions and comments and pics to TWiV@microbe.tv. And if you enjoy these programs, we'd love your support. Microbe.tv/contribute. Angela Mingarelli is at McGill University Immune Vet on Blue Sky. Thank you, Angela.
Thank you. I had a great time.
Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Keep looking up. Keep looking down.
Into the microscope.
Yeah, that's what I was thinking. Keep looking down.
I know. Hopefully everyone caught that.